Assalamu alaikum dear students, hope you are fine by the grace of Almighty Allah. Welcome to NIVS Home School. I am Sabreena, Haque, assistant teacher of Science, National Ideal English Version School, Day Shift. Dear students, today I am going to take a biology class for class 9 student on the based on chapter 14. This is our part 2 of the chapter, and along with the part 2 we will discuss the creative question of these chapters which is belongs to the exercise. So let us move forward to today's topic. The name of the chapter is biotechnology. Today we will start 14 decimal 3.1 the stages of the preparation of GMO or DNA recombination. What is GMO? The GMO is genetically modified organism. In the human dietary canal there lives a type of bacteria that is known as E. coli. Researching on this bacteria, most of the technique of genetic engineering has been developed. Genetic engineering or recombinant DNA technique is accomplished by the steps mentioned below. First step will be DNA with desired gene is separated from the donor organism, then plasmid DNA is separated from the bacteria to use it as a carrier of the gene. Plasmid is an individual DNA besides the chromosome in the bacteria which is able to divide or able in self division. Now come to next one. In this step the plasmid DNA and donor DNA are divided by a special enzyme. Desired gene is present in any of the part, that means location of the donor DNA. And question part next step is then donor DNA is placed between the two terminal parts of the a plasmid by lipase enzyme. Lipase here act as adhesive. Now as a result DNA recombinant form and with the specific desired gene. This recombinant plasmid now carries the desired part of the donor. And next step is in this stage the bacteria recombinant DNA emerges into receiver bacteria. The method of emergence of donor chopped DNA part into bacteria is called transgenic organism. A new species of bacteria or organism created due to transformation is called transgenic organism. Next step is in this stage bacteria with recombinant plasmid are identified and then separated. Bacteria with the desired gene are made reproduce rapidly. Now each of these reproduced bacteria contain the desired gene. The process by which gene is reproduced is called the gene cloning, and to use the gene plasmid is separated again. Now come to picture of these steps. This is the E. coli bacteria, and from our desired animals or plants we have to cut down the desired gene by the restriction enzyme. The cutting of DNA with restriction enzyme, and with this part, that desired gene part, we have to which plasmid we take from the E. coli bacteria, that part will be stuck with this plasmid with the help of lipase. Here lipase helps to join this. Here development of recombinant DNA adhesion with the both DNA and DNA ligase. Then desired gene is here with the ligase. It is the recombinant DNA plasmid. This plasmid is inserted recombinant DNA plasmid in bacteria through the transformation, and now the cloning of the bacteria is happening here, and through the cloning we will get so many desired gene from the many bacteria. Now come to the next page. This technology is being significantly used by innovators or investors because in the short period of time very accurately desired characteristics can be transferred by transferring the gene. Genetic engineering is effective for the development of new crops in the comparison with the traditional ways of reproduction, because the transfer of gene is similar or limited to the nearest species through traditional reproduction, but it is possible to directly transfer one or more gene to nearer or distant species through genetic engineering. Moreover, it required a long time to achieve desired result through the traditional way of reproduction. It is possible to obtain plants and or animals or microorganisms with desired characteristics in a very short period of time with genetic engineering. In case of traditional reproduction, unexpected genes may transfer with the desired gene, and the successful transfer of the desired gene remains uncertain. In genetic engineering, there is no possibility of transferring unexpected genes, and the transfer of desired gene is certain. Traditional reproduction is not controlled by any rules or method of biosafety, but in the case of genetic engineering, it is controlled by internationally accredited rules and methods of biosafety. Toxicity is not tested in traditional reproduction, but is done in genetic engineering. Now come to uses of genetic engineering. Genetic engineering or recombinant DNA technology is most modern biotechnology. The main objective of the technology is to develop organisms with new and advanced characteristics, and by which human being can be benefited. By this time, notable successes have been already achieved through this technology. Now come to first use in the developing crops. Insect resistance varieties of the crops have been developed, such as Bt corn, Bt cotton, Bt rice, that is developed in China. These crops are resistant to harmful insects under the Lepidoptera in the Coleoptera orders. Using this technology, virus resistance varieties of crops have been developed, and for the intents of transferring the gene tomato mosaic virus, tobacco mosaic virus, and tobacco mild green mosaic virus resistance varieties of crops have been developed. The variety of papaya that is resistant to ring spot virus and has also been developed. Research is being carried out for the development of the light resistance of the lead blight resistance variety of the potato by the transferring of lead blight fungus resistance gene. Dear students, here all the name of the disease and all the name of the characteristics which is developed by using the genetic engineering you have to learn. This is very important for your MCQ. And varieties of corn and cotton tolerant to herbicides have been produced by genetic modification. Now scientists have become able to develop herbicide tolerant tomatoes by transferring herbicide tolerant gene into tomato from the bacteria. This way, herbicide tolerant varieties of soybean, corn, cotton, canola, etc. have been produced. Through genetic engineering, more than one trait can be inserted into same plants. Nowadays, this type of transgenic plant is commercially available for intents side by side. Both herbicide tolerant and insect resistant characteristics have been inserted in corn and cotton. Through genetic change, the nutrition value of some crops have been improved. For example, vitamin A beta carotene gene has been transferred into rice. Effort to add iron to the rice are being continued. Attempts are also made to develop salinity and drought resistance varieties of the crops through the genetic modification. Now come to in animal. In livestock, for example, transfer of protein C gene has been done to increase the protein of cow milk, and though this is still in research level. Through genetic modification, genetic changes have been accomplished with the sheep by transferring the growth hormone producing gene of the human being to it, with the view of increasing its size and meat production to improve the amount and the quality of sheep's fur. Two bacterial genes CSE and CSM have been transferred to the genome of the sheep. Now come to the improvement of fishing. Transfer of growth hormone gene of salmon fish through genetic modification to catfish, common carp, Loitea nilotica can increase the growth rate of the fishes by 60%. Now come to the healthcare. The hepatitis B virus vaccine is being produced from the yeast through genetic modification from genetically modified E. coli bacteria and yeast, insulin is being commercially produced for the treatment of diabetes by using the gene which produces insulin in the human body. The human growth hormone and component of the stimulant for the acceleration growth of the granulocytes macrophage colony are being produced from the genetically modified E. coli bacteria and yeast, and these are being used in dwarfism, then viral disease and cancer AIDS. Now last point here, for environment protection, the areas of petroleum industry and refinery of coal mining are being free from pollution because this technology is being used to make the environment management easy and fast, such as management of industrial wastage and so as Dr. M.K. Chakraborty, by performing research on genetic engineering, has developed a variety of Pseudomonas bacteria which is able to disintegrate oil and hydrocarbon in any environment. Dear students, now we will solve the MCQ of the exercise. First MCQ, which one is the special enzyme to cut the DNA? Ligase, lactase, restriction and lipase. Dear students, here restriction is the right answer to cut the special enzyme is to cut the DNA, and lipase is used for joining. Now come to biotechnology is applied to fermentation, tissue culture, develop transgenic organism. All are correct. So answer will be D. Now read the following stem and answer the question 3 and 4. Imtiaz found a very good variety of wood apple. Visiting his friend's house to produce the plantlets with the same characteristics, he brought some lateral bursts of the plants and produced them in the university lab and of the botany department. The first question, this will be question number 3 and this will be question number 4. The question 3 is, what is the process that Imtiaz followed in the lab? Transfer of gene, use of enzyme, application of hormone, tissue culture. That will be tissue culture. Through the tissue culture we can produce a lots of plantlets in the short period of time. The next question is, which one shows the consecutive steps of Imtiaz activities? Dear students, we know the consecutive steps of the tissue culture is selection of mother plant, preparation of culture media, then inoculation of explant, development of plantlets and transfer to natural environment. The C has all the points properly, so answer will be C. You have to put tick mark on C. Now we will discuss on creative question 1, that is your exercise creative question. In the garden belonging the genetic engineer Dr. Haidar, the lemon plants are dying rapidly from the disease. Through the lemon harvest is the high. He finds that there are some lemon trees of the same species which lives for a long time, although they do not produce high yield. From these two varieties of lemon, he developed a new high yielding and disease resistant variety. He produced the plantlets not using the traditional process but by a special process in his lab. First question, what is biotechnology? Second question, what is GMO? Third question, explain the technique followed by Dr. Haidar for the development of lemon variety. And fourth question, explain the reason of developing plantlets through the special process by Dr. Haidar. The answer is A. Biotechnology is the use of biological process, microorganism or system to develop any new organism with new character intended to improve human life. Now next question was, what is GMO? The acronym GMO stands for the genetically modified organism. To develop new characters changing in DNA of any organism is genetic engineering. With this technology, the transfer of the desired part of DNA from bacteria to human being, from plants to animals and from animals to plants have become possible now. The organism with new character is called GMO. Now come to question answer C. Dr. Haidar followed the following technique for the development of lemon variety. First one, selection of targeted DNA. Second one, selection of carriers so that transfer of desired segment of DNA become possible. Third one, selection of necessary restriction enzyme to chop the DNA molecule at the particular locus. And fourth one, selection of DNA ligase enzyme to join the segment of DNA chopped. And fifth one, selection of the host for the replication of carrier DNA with the segment of desired DNA. Sixth one, evaluation of the expression of the recombinant DNA prepared with desired DNA segment. Now come to question answer of D. Now the question is, nowadays in the reproduction of plants and in the field of developing new variety, much achievement has been obtained by utilizing the tissue culture. Dr. Haque develops new lemon plants with new characters by genetic engineering and produce plantlets by tissue culture process. Dr. Haque develops his plantlets through the tissue culture process, because first one, innumerable plants can be produced with the same character by this process in a short period of time. Second one, disease free and especially viruses free plantlets can be produced easily. Third one, it can be free from limitation of producing plantlets in the specific season. Fourth one, sufficient number of plants can be produced and the problem of the starting of seed can be avoided. And fifth one, rapid multiplication of plants which do not reproduce cell through sexual reproduction can be done by culturing embryo. And last one, they can be speedily transferred into a short period of time in fresh condition. Dear students, this is your homework number 18. You have to write CQ 1, C and D as your homework, and you have to submit on Saturday from 9 am to 3 pm. Dear students, I have finished my today's topic. Hope you have understood. Read the chapter properly and do the SW. Thank you. Allah Hafiz.